All right, Chapter 10 is on classification of microorganisms. We've already touched upon this just a little bit in our introduction chapter, where we talked about what a microorganism is and kind of talked about the different domains and the different groups, but we're just going to take a little bit more time, but mainly focus on ways that we can identify and classify organisms. So we're going to look at differentiating between taxonomy and phylogeny. Look again at the three domains of living things, list characteristics of the three domains, and then look at the various methods for classifying and identifying organisms. Some of them we've already used in lab, so they should sound familiar to you. So first of all, we want to distinguish between taxonomy and phylogeny. Taxonomy is the science of classifying organisms. So what category are we going to put this newly discovered organism into? And so taxa are those categories of organisms. This is not to be confused with taxidermy, which is the stuffing of dead animals. Okay? All right. Phylogeny is the study of the evolutionary history of organisms. So when we look at how we organize the various categories of things, that hierarchy, that kind of reflects some of the evolutionary relationships. So the two are related, but phylogeny looks more at how do we kind of separate out the organisms based on common ancestry and how they may have evolved over time. So again, a review of the three domains. We have the archaea, ancient bacteria. Okay, archaea, so archaic is old, okay? So these are prokaryotes, meaning no nucleus, okay? They do not have peptidoglycan, okay? That's what distinguishes them from our everyday bacteria. These are the ones that can live in the very extreme environments, so there can be like the methanogens, the extreme halophiles, the hyperthermophiles that live in like volcanic vents that are really hot, those hot springs, places like that. The bacteria, which is what you're more familiar with, this is your everyday bacteria that if you get a bacterial infection, bacteria that live in and on you, in your gut and so on your skin, that belongs to this category, this domain. They are, again, prokaryotes. They have cell walls with peptidoglycan. And you know that because your gram-positives have a thick peptidoglycan layer, your gram-negatives have a thin peptidoglycan layer. They could be both pathogenic and non-pathogenic, meaning they can be, pathogenic means disease-causing. Okay? And then there can be non-disease-causing ones as well. Everything else is the eukarya, meaning they are eukaryotes. They have a nucleus. Okay, so everything else. So we often talk about these as kingdom Animalia, kingdom Plantae, kingdom Fungi, kingdom Protista. Okay, so the rest of these. You know, animals, they're the ones that can ingest food to take it in. Plants can make their own food. They're autotrophs, make their food through photosynthesis. Fungi absorb their food through tiny root-like structures called hyphae. And protists, they're kind of the catch-all kingdom that they could have like the plant-like protists that are the algae, or they can have the animal-like protists, which we call the protozoa. And they have lots of different ways of swimming around and moving around and so on. So when we talk about phylogenetic hierarchy, we're grouping organisms according to common properties. So what it basically implies when we do that is that those organisms evolved from a common ancestor. And from that, we can make things like phylogenetic trees to show how each organism's branched off. We can make cladograms and so on. We've used fossils to help with organizing this hierarchy of when organisms evolved. But more recently, we can use DNA evidence to see how related different organisms are to each other. Once we have identified a new organism and maybe have decided which category where we're going to classify it, we do have to give it a name. And we use what's called binomial, bi equals 2, name, nomenclature. That's your word naming system. This was first developed by Carlos Linnaeus. And it's basically composed of your genus and species name. So the first word is always the genus, and it's always capitalized. The second word is the species, and it's always lowercase. We write it in italics if you're typing it, or you can underline it if you're just handwriting it out. And the reason the scientific names sound so weird is that they're often written in Latin. So, Escherichia coli. I would have to underline it because I can't write in italics, even though my writing looks sloppy like it is. But this would be your genus name right here, and species. Okay. When we look at, a lot of times, you know, maybe in biology and stuff, we started talking about kingdom, phylum, class over order, family, genus, species. King Philip came over from Germany Saturday. That's how I remembered it and was taught it in biology. But what we've added now is that top domain. And so the domain is either going to be eukarya, bacteria, or archaea. And then from there, we can add the various other taxonomic hierarchies when we're classifying an organism. Now, when we look at viruses, this is a little bit different because we do not classify in the 3 domains. Why is that? They're acellular. They are not made of cells. All other organisms are at least unicellular, like the archaea and the bacteria, or they're multicellular, but they're made of cells. Viruses are not. Viruses cannot survive on their own. They have to take over host cell machinery to help replicate and make more of themselves. So they're generally more closely related to their host that they invade than they are other viruses. A viral species, we consider that a population of viruses with similar characteristics that occupy a particular ecological niche. And there's 2 hypotheses as how viruses came about. One hypothesis is that they arose independently from replicating strands of nucleic acids, maybe from a plasmid that developed an outer protein coat and eventually was able to invade other organisms. Or they developed from some degenerative cells that basically lost their ability to survive independently. So maybe a simple organism that just lost its ability to survive on its own. As far as classifying and identifying microorganisms, there are a lot of different methods. A lot of them now involve a lot of DNA technology. So I don't even know all the specifics about them because they're very new. But we've already done some of this in lab. Morphological characteristics, that is looking at the size, shape, arrangement, you know, is yours a gram positive, gram negative? I mean, is it a coccus, spirillum, a bacillus? Is it a form tetrads, chains, and so on? Differential staining would be like your gram stain. Biochemical tests would be like your phenol red tubes. Some of your other augurs that are differential, that show whether or not an organism, you know, metabolizes a certain type of sugar in there. Serology looks at your immune responses because we produce antibodies in response to microorganisms. So anytime they have on the surface of their cell proteins called antigens, and anytime we're subjected to these foreign antigens, we build up antibodies against it. So serology looks at if you've got antibodies built up against something, that means that that particular thing has invaded you. And that's even what they've done with COVID, is that they check to see those individuals if they are producing antibodies against that virus. Phage typing looks at different bacteriophages, bacterium's susceptibility to being invaded by a microphage, by a bacteriophage. Okay, that's a virus that invades bacteria. Okay. They look at different fatty acids that bacteria produce to see how closely related they are, if they produce similar fatty acids. Flow cytometry is used for analyzing liquids. You know, if they want to see if a particular liquid has microbes in it, maybe they are checking milk to make sure it hasn't been contaminated with a certain bacterium, or if they're checking a town's water supply to see if it has E. coli in it, they might use flow cytometry because they can have that liquid flow through the machinery to detect levels of that bacteria. DNA base composition, as the name suggests, they're going to look to see, do they have a similar sequence of bases as another organism over here. DNA fingerprinting uses restriction enzymes. To cut DNA at certain spaces, at certain spots. What that will generate is different sized fragments. If they're looking at a certain sequence AACCGG, if they're going to cut it at that spot in different organisms' DNA, where they find that, all organisms might have that sequence, but they might have it in a slightly different spot. So each time that's cut, it will generate different sized pieces. Then we put that into a gel electrophoresis and let those get separated, and they show up as banding patterns, and then you look to see which banding patterns look similar. You would assume those organisms are more closely related then. Polymerase chain reaction, or PCR, is just a method of rapidly amplifying that we just mean copying DNA. So microbes aren't very big, so if we need to get more of their DNA to analyze it to see how closely related they are, we would just pop it into a thermocycler and just keep rapidly dividing, making more copies of it so that we could get enough DNA to look at. Nucleic acid hybridization, they want to see how closely two different organisms, their DNA could join. So let's say when DNA gets separated during DNA replication into its separate strands, let's say you had another organism and you did the same thing. You got some helicase and separated the DNA as two strands. If those two organisms' DNA could fit together, join together as if it was just one molecule, that means they must have pretty similar series of DNA bases for them to be able to join together segments of their DNA. They must have really similar sequences. So if those sequences are similar, then it means that they are probably more closely related as well. All right, so the last little bit is on methods we use for classifying. So once we've figured out the characteristics, maybe the DNA patterns, different things about an organism, we might use some methods to just classify them and get them into the right taxa or category. And so dichotomous keys, di means two, dichotomous keys give you successive questions with two possible answers. Does it have this or this? Is it a prokaryote? Yes or no? I mean, there's always two choices only. And you keep using those questions until you've basically keyed out or figured out what your organism is or how to classify it. A cladogram shows evolutionary relationships. And so each branch point on the cladogram is a feature shared by a species on that branch and will relate to a common ancestor. And we'll show you in class how we can figure those out, but you've got a page on 294 that kind of shows you that. Oops, wrong thing.